will be Professor Sara Liin from Linköping University. She's going to be talking to us about modulation of wild type and mutant cardiac Kv7.1, KCNE1, potassium channels by lipid molecules. There you go. And your presentation's up as well, Sara. All running perfectly in the background. I'll let you get on with it. Over to you, Sara.
Thank you very much. I hope you all can hear me, and otherwise just shout. So I'm Sara. And first of all, thank you for the very kind invitation of being part of this exciting symposia of today. It's a pleasure being one of the speakers. So I will today present some of our kind of recent and ongoing work on how different lipid molecules are affecting the cardiac Kv7.1 KCNE1 channel. And I will refrain from going into too detailed biophysics details, and instead I will kind of describe the rationale why we're interested in this channel, why we are interested in a particular class of lipid molecules, and I will end by describing some of the ongoing questions that we're trying to address right now, and maybe you have some good input there. But I would be happy to discuss more details afterwards. So let's see. So why do we want to target the cardiac Kv7.1 KCNE1 channel? Well, this is a channel, a voltage-gated potassium channel, and belongs to the Kv7 subfamily, which has 5 members. And we just heard a very exciting talk by Bernard about the neuronal counterparts, Kv7.2 and 7.3, whereas my talk will move to the heart, where Kv7.1 is most famous for its role in cardiomyocytes, ventricular cardiomyocytes, where it co-assembles with the KCNE1 subunit to form a channel complex called Kv7.1 KCNE1. And this channel is generating a repolarizing potassium current in the heart, and it's a slow potassium current, or slowly developing potassium current, and that's why many are referring to this channel as the IKS channel. So just a quick reminder, the Kv7 channels, they are typical voltage-gated potassium channels. So to the left here, you see a top view of the channel architecture with a central ion-conducting pore domain flanked by 4 voltage sensing domain. And to the right, you have a very schematic illustration of how conformational changes in the channel that are triggered upon depolarization. It triggers the outward movement of the voltage sensor, which is marked by positive signs in the protein here. And this upward S4 movement is coupled to the pore domain to allow for opening of the ion-conducting pathway and allow potassium ions to leave the cardiomyocyte. And one reason why we are so interested in the Kv7.1 KCNE1 channel is its link to cardiac arrhythmias and its important role for cardiomyocyte repolarization. So the IKS channel, or the Kv7.1 KCNE1 channel, it contributes to the repolarizing phase and is very important for the timing of repolarization. So if this channel is failing, for instance, because of loss of function mutations, you tend to get a prolonged action potential duration, which clinically is detected as a prolonged QT interval. So that's why it's linked to an arrhythmia called long QT syndrome. And this prolonged QT interval, it predisposes the heart to early after depolarization and ventricular arrhythmias, and in worst case, sudden cardiac death. So we're fascinated by how the channel normally works, what goes wrong when you inherit mutations in the channel, and how you can restore the function of loss of function mutants. And to the right, we see an example from the Roden lab where they have plotted different residues in the channel that are linked to inherited forms of long QT syndrome. So there are hundreds of point mutations in the Kv7.1 KCNE1 channel that in different ways cause loss of function, and are suggested to cause the arrhythmias in these individuals carrying the mutations. So the overall aim of much of the work that we do in the lab is to develop or identify compounds that activate the Kv7.1 casein E1 channel. And we're not the only ones doing this. I think this is a general effort from many different groups and different research teams. But the idea of identifying and developing Kv7.1 casein E1 activators is that restoring or augmenting the activity of this channel would bring back a well-timed cardiomyocyte repolarization, which in turn would bring back a normal QT interval. So I will describe today some of the work we've done in this overall project. And we have been focused on different lipid molecules. And just briefly, why are we interested in lipid molecules? Well, we know that all the Kv7 channels like to interact with lipids. I think the most famous examples is what we see to the left here, which is the phospholipid PIP2, which has been studied by many groups, and here it's detected in a cryo-EM structure of this particular channel. And it's a very important modulator of channel function. But it's also known from work of many different groups that also other type of lipids regulate the function of Kv7 channels and other voltage-gated potassium channels. For instance, pollen-saturated fatty acids, or shortly PUFAs, that we see to the right here, they interact with many different voltage-gated channels to modulate their activity. And we were interested in, would Kv7.1 casein E1 also be modulated by PUFAs or similar compounds? And just to give a heads up here that I have a conflict of interest by a patent application, so I'm very fond of lipids, and it could be good to know. Most of the data you will see, it's from manual electrophysiology. I saw the poll that there were a roughly 50-50 distribution of the audience, and we primarily work with the xenopus oocyte system, where we express human Kv7.1 casein E1 to generate currents typical of what you see in the middle panel here, where we can look at current amplitude, we can look at opening and closing kinetics, but we can also plot the voltage dependence of the channels, perfuse in different fatty acid molecules, and see how the activity of the channel is changing. And just to keep in mind, what we're looking for in this project are Kv7.1 casein E1 activators. So this is an example of the early work we've done studying PUFAs or PUFA analogs on the Kv7.1 casein E1 channel. And here you see one of our first favorite compounds. It is a PUFA analog by having a pollen-saturated arachidonic acid tail, but it has a synthetic head group, a taurine head group. And what's special with this head group is that it's very good at keeping a negative charge. And this is very important for electrostatic interactions with the channel, and we'll come back to that later. And this is quite similar to the properties needed, for instance, for PIP2 to interact with different channels. And I think you can appreciate already from this slide that if you compare the black and the red lines here, this PUFA analog NAT, it's a quite good activator of the channel. So it's increasing the current at depolarizing the stimuli, like you see in the left. And it's doing this by shifting the voltage dependence of channel opening towards more negative voltages, but it's also increasing the maximum conductance at the most positive voltages. So we like oocytes, and we work mainly in oocytes, but we were initially interested in seeing if these effects can be detected in other type of experimental models as well. And just briefly to highlight some of these experiments that we've done in other models. So to the left, you see embryonic rat cardiomyocytes, which are first treated with an IKS channel blocker to induce arrhythmic beating. And when we then co-apply together with the blocker this PUFA analog NAT, we found that NAT is quite good at restoring rhythmic beating of these cardiomyocytes. To the right, you instead see experiments from isolated guinea pig hearts, where we've done different type of experiments, first to prolong action potential duration or QT interval, and this is to mimic long QT syndrome, and it can be done with hERG blockers or IKS blockers. And in this experimental setting, we found that, for instance, NAT is good at restoring a baseline action potential duration in QT interval. So this was very encouraging. We have a PUFA analog which is activating the channel, and it has nice effects not only in oocytes but also in more complex models. The kind of backside of this initial story was that we need quite high concentration of this PUFA analog to have beneficial effect. So in our hands, we need to use about 30 micromolar or 70 micromolar to get the effects we like. So we continued studying these effects and the mechanism of action to figure out how can we improve these PUFA analogs, how can we improve their chemistry, their interaction with the channel, and to do that we need to know how they interact with the channel. And this has been long-term detective work, so I will just give you kind of the essence of what we found, but I'm happy to discuss later on. But through molecular dynamics and metagenesis and also electrophysiology work, we found that PUFAs and PUFA analogs, they have two main interaction sites on the Kv7.1 channel that underlie the activating effect. So one site, which is marked by 1 here, it's at the voltage sensing domain, and it's characterized by the negative head group of these molecules interacting with the voltage sensor of the channel to facilitate its outward movement, and in this way help activating the channel. Another site marked by 2 here, it's closer to the pore domain. It's also electrostatic interactions, but with a positive residue behind the selectivity filter, and this helps coordinate the potassium conductance through the pore of the channel. And in this platform, we're not really, it's not possible to show movies, so you have to believe me when I say that in molecular dynamics, these lipid molecules are attaching to the channel nicely electrostatically with the head groups, whereas the blue tail here is kind of dancing around the channel and making many hydrophobic interactions with a network of hydrophobic pockets on the channel protein. So from this type of work, we learned what's important in the PUFA analog molecule to be able to interact with the Kv7.1 casein E1 channel. In the next step, we tried to improve this interaction, and this was done by synthetic chemistry, where we play with the different components of these lipid molecules. So by combining different head groups with different properties and geometry and different charge with different tails, we created a small library of PUFA analogs with good effects on the channel. And one thing we noticed when we made these combinations was that several of the PUFA analogs that were good activators of Kv7.1 casein E1, they actually belong to an endogenous family of lipid molecules, which are called the endocannabinoids. So in our more recent and ongoing work, which is still preliminary data, we have been building on this efforts with synthetic lipids and instead studying how endogenous endocannabinoids are activating the Kv7.1 casein E1 channel. And just to give an example, so here is a very small manual screen we done on the human Kv7.1 casein E1 channel in xenopus oocytes, where we've been testing the effect of some of the more famous endocannabinoids, the 2-AG and anandamide, but also some of the more unstudied and kind of newly discovered endocannabinoid compounds like RAS and DOCS, which are fatty acids linked to different type of head groups like serine head groups or GABA head groups. And in our hands, we found that typical endocannabinoids like 2-AG and anandamide, they are not activating the Kv7.1 casein E1 channel. So these are the ones that are blue or gray in these different bars. But then you have all the red ones, which are the less studied endocannabinoid compounds, and they are actually very good activators of the Kv7.1 casein E1 channel. They are even better than many of the ones that we try to develop ourselves using synthetic chemistry. So you see this activating effect. So here we're using a concentration of 10 micromolar, and many of these, they shift the voltage dependence of activation about 40 to 50 millivolt towards more negative voltages. And to the right, you see that they are good at increasing the steady state current at different voltages that kind of corresponds more to the plateau phase of the cardiomyocyte action potential. One of our favorite endocannabinoid compounds is called RAS, and it's not only because it reads Sara backwards, but it's because it's based on an arachidonic acid tail, and we know that there are a lot of arachidonic acid in the membrane of human cells, so this is anticipated to be one of the more abundant endocannabinoid compounds of the unusual ones. And here you see an example of 10 micromolar of RAS, what effect it has on the Kv7.1 casein E1 current. So it's increasing the overall current, but it's also shifting the voltage dependence towards negative voltages. It's doing this in a concentration dependent manner with an apparent affinity of about 5 to 8 micromolar. So it works much better than our initial favorite, which was NAT. So we can now titrate down the concentration to get beneficial effect of this endocannabinoid compound. And one reason why we're so interested in the RAS effect is that it goes exactly opposite to what many of the long QT associated mutations do. So if we now expand on this figure that was in a very nice study from the Rodin lab, what they did plot here is how much different mutations linked to arrhythmia are shifting the voltage dependence of channel activation towards more positive voltages. So dark color is a bigger shift. And to the right, you see an example from our own of what one of the mutants is doing, and it is shifting the voltage dependence towards more positive voltages, which also gives smaller current at different voltages. So in the next step, and this is now very preliminary data and ongoing work, we're looking at how good RAS is to shift back the voltage dependence of such mutations. So here an example of some of the mutants we studied so far, and all of these are point mutations found in patients, and they're linked to long QT syndrome. And what's encouraging to find is that fairly low concentrations of RAS, so 3 and 10 micromolar, is shifting the voltage dependence of all these mutants towards more negative voltages. And you see ranges from -10 to up to -50 millivolt shift. The bottom graphs shows that RAS is increasing the current, the steady state current, in this case at 0 millivolt. So we think it's nice to see that the effect of RAS goes opposite to what the mutants, the defect of the mutant is. One thing I like to highlight that we're studying in more detail right now is that not all mutants respond to the same extent. So to the very left, you have the I204F mutant, which in some parameters respond less than wild type and some other mutants. Whereas to the very right, you have the A370V mutant, which is responding very nicely in some parameters. So it seems to be the more sensitive to RAS effects. And this is something we'd like to study in more detail. So if I summarize what we had so far, so we've seen that both synthetic and endogenous proof analogs are acting as Kv7.1 casein E1 activators. I didn't go into detail, but we have identified 2 sites of action on the channel and pinpointed what these interaction sites look like and what the mechanisms are behind the activating effect. We found good activators among endocannabinoids, and we found that some of these endocannabinoids, like RAS, it's augmenting the function both of wild-type channels, but also of Kv7.1 casein E1 channels that harbor arrhythmia-linked mutations. So we like the endocannabinoids, and we're interested in studying them further. And one of the aspects we're really interested in is, could these be putative endogenous modulators of this channel? And is this something we can use in drug development to tweak the chemistry or to develop other compounds that are using these sites and the mechanisms of action? What keeps us busy right now is that we're continuing this preliminary work I showed on the impact of long QT mutants. So which long QT mutants respond well to endocannabinoids, and are there diverse responses, and how could that come in such case? Something that might come from the blue here is that we're also interested in the impact of the agenetic state of the channel, because this channel is mainly important during kind of heart fast frequency beats of the heart, such as during agenetic stress. So we were guided by nice work by the FEDIDA lab, who showed that a double mutant is mimicking the phosphorylation of the channel that is kind of triggered by beta agenetic stimulation. And we are surprised, we don't know the reason yet, but it seems like this phosphorylation mimetic mutant is responding better to RAS than the wild type channel. And this is something we like to follow up and also understand. Another aspect I'd like to end with is, of course, we like to know the translational potential of what we do. So we mainly work in oocytes to get to the mechanistic details, but how do our effects translate to mammalian cell lines and to cardiomyocyte, to whole hearts, and even more complex systems? We've done that to some extent using guinea pig models, for instance, but it's something we like to extend for the endocannabinoids. And that's why I'm so excited that just opposite here in the corridor, we're just getting help to install and calibrate a Q-patch that we'll be using for the coming years. I'm really excited with the guidance from Sophion, and this is hosted by the Linder lab, that we can hopefully repeat many of the experiments in cell lines and also expand on what happens on other type of cardiac ion channels. So with this, I like to acknowledge past and present members in my lab, and also the synthetic platform that we're collaborating with. Also collaborators at University of Miami and in Calgary and in Copenhagen. So this is a team effort using different models to study these endocannabinoid and proof analog compounds. And I'm happy to take more questions if you have on the mechanistic details. Otherwise, I hope to get back at another time point with learning what a Q-patch could help us with. Thank you very much.
Lovely, Sara. Thank you very much. I remember sort of seeing some work on this from your lab and from other labs from University of Manchester, I believe, some years ago, and I wish I'd kind of pursued it a bit more because it's really fascinating stuff. But anyway, I can't keep you too long because we've got to move on, and there's plenty of good questions for you. So, Savi Gentili, do you have data about the potential effects of these compounds on non-excitable cells? And I think this is basically a two-part question because he goes on to talk about, in a therapeutic environment, for instance, you might be administering these compounds systemically. So could you expand on the potential side effects on, other ion channels, for instance?
Yeah, exactly. Yeah, it's a good question. And I must say that no, the brief answer is no, we don't know. We hope that we will at least be able to address within the coming years what effects, for instance, RAS has on other cardiac ion channels. I would say this is the next step, because in the past we tried some of the more synthetic PUFA analogs and they have surprised us. Some are very specific for this channel, and some have broad effects also on sodium and calcium channels. So it's hard at this point to predict. We have to test it. But otherwise, I would say that endocannabinoids are tricky because they do a lot of different things. What's good with RAS is that it has a very poor affinity for cannabinoid receptors. So I don't expect adverse effects through that signaling system. But otherwise, I don't know. And that's why we're looking into other molecules using the same mechanism, but not being endocannabinoids in themselves.
And another question here from Bernard Attali. Why do endocannabinoids have a voltage-dependent effect? They appear to be more active at 0 millivolts versus plus 40 millivolts, for instance.
Yeah, exactly. And I guess, well, we probably need to know better exactly how the endocannabinoids work. And this is something we're working on. But I would say that probably it has to do with that they have two effects on the channel. So they have one effect on voltage sensor movement, which contributes to shift in V50. But it seems to have more binding side, and one is at a pore to stabilize somehow the conducting selectivity filter. So maybe at some voltages that these two effects add together more than they do at other voltages where the channel already kind of reached saturation in its voltage dependent opening.
And I think one last question, we've probably got a few more, but I think we have to sort of try and call it a day at some point. But one last question from Brooke. Do you know if endocannabinoids have side effects on other ion channels within cardiomyocytes, and do they affect other aspects of cardiac function, such as calcium handling?
Calcium handling, I don't know, but I know that they have effects, I think, on some TRIP channels, for instance, and they have been shown in kind of a more complex tissue, for instance, to cause vascular relaxation, which could affect kind of the perfusion and the blood flow through cardiac vasculature as well. But I think we're just starting to put together the different pieces of what could be a net effect. I think there are many channels that are affected by these ones, but some are more selective than others, and we need to do that kind of mapping of this to know exactly which one has which type of profile.
Great, thanks, Sara. That's lovely talk and some really nice answers and questions there from the audience.